Excuse me, Dr. Anna, you're in mute.
Okay, so going back. Welcome everyone to this new chapter of our webinar series on MicrobeTalks, Exploring the Microbial World. This is a series of seminars sponsored by the Puerto Rico Society for Microbiology. Our guest today is Dr. Aarti Raja. She earned her PhD in microbiology from the University of Maryland in 2000 and completed postdoctoral training at Harvard Medical School. She joined Nova Southeastern University in 2012 as a faculty member and now holds the rank of professor in the Department of Biological Sciences. Her primary research focuses on microbiology with a specialization in virology, particularly the human immunodeficiency virus, HIV, and its associated proteins. With over 12 years of virology research experience, Dr. Raja has significantly contributed to understanding infectious diseases and their implications. Beyond her laboratory work, she is a passionate advocate for innovation in science education, creating opportunities for students to explore research early in their academic journeys. Dr. Raja actively participates in global scientific collaborations, notably through the Tiny Earth Project, which empowers undergraduate students to engage in groundbreaking research to address antibiotic resistance. At Nova Southeastern University, she co-leads Science Alive, an initiative that fosters connections between the university and local schools, bringing interactive science education to children and inspiring the next generation of scientists. In addition to her academic and research pursuits, Dr. Raja has extensive experience consulting on infectious diseases for pharmaceutical and financial industries. Her expertise bridges scientific discovery, education, and real-world application, showcasing her dedication to advancing both research and public understanding of science. So welcome, Dr. Raja, and your lecture entitled Tiny Earth, a Cure to Address Global Challenges.
Thank you so much, Dr. Anna. It's a pleasure to be here, and thank you very much for inviting me to be a part of this group and to speak with you all. So can I go ahead and start?
Yes.
Okay, great. So it's my pleasure to be here to speak about Tiny Earth, which is what we call a cure to address global challenges. A cure specifically stands for a course-based undergraduate research experience. And I will speak about how Tiny Earth works and share some of that information with you all here today. I just want to apologize in advance. I'm battling a little bit of a cold, so excuse me if I cough in the middle. And I also have my little pet dog here, so he might bark occasionally, so excuse me for that. So as being part of Tiny Earth, one of the things that we do at Tiny Earth is we always begin by providing what we call a land acknowledgement. And so I'd like to do the same here as I begin my talk. This is just a map of where I am situated, which is South Florida. I am about 35 minutes north of Miami. And what this map is just showing you is the original population of people who inhabited these lands. And so I'd just like to read a little bit about the land acknowledgement that we like to offer before we begin any work here regarding Tiny Earth in South Florida. So many of today's participating institutions in Tiny Earth, they actually occupy, conduct research on and actually source soil from many ancestral tribal lands. So Nova Southeastern University, where I am currently, recognizes that we reside on these ancestral lands of indigenous people who actually still occupy these territories. So we wish to especially acknowledge the connection of these communities, their elders, and future generations to this land. So just to give a brief history of this land here. So many dozens of Florida Indian cultures flourished in this environment well before the Spanish arrived onto this land. And over time, unfortunately, with movement of people from Europe to the lands in Florida, they also brought with them a lot of deadly diseases, which took away a lot of the Indian population. Eventually, when Florida got relinquished over to Britain, many of the local population actually left this area. And so, in fact, a lot of the Florida original Indians just weren't here anymore. The indigenous native population was gone. But with that came that vacuum, and as a result, there was a lot of movement of people or tribal people from different parts of the United States, primarily Georgia, Alabama, and some other regions. And these were known as the Seminole Indians and the Miccosukee Indians. So these are the people, the Seminoles and the Miccosukee Indians were the people who lived in Florida when this became part of the United States. And over time, unfortunately, there were a lot of conflicts when the United States was becoming settled by many people. And the conflicts led to a lot of Indians being captured and surrendered, or they just ended up moving west. But there were left behind a few hundred Indians who occupied the region of the Everglades, the Big Cypress Swamp, and the 10,000 islands around the state of Florida. It is these descendants of the unconquered Seminole and Miccosukee tribes who live here today in Florida. We recognize the tremendous loss, forced removal, and unethical assimilation that has resulted from global colonization of indigenous peoples. This history is not isolated to a single tribal nation, but it informs our shared future of collaboration and healing, while honoring the sacred traditions and culture that continue to persevere. So what is Tiny Earth? So Tiny Earth is actually a global network of faculty members or instructors and students, and we refer to this word as students sourcing antibiotics from soil bacteria. I've written mostly college because traditionally Tiny Earth as a project was designed to run in college classrooms, but I can speak later about examples where it also works in a high school setting. So Tiny Earth actually emerged from a course that was originally designed, created, and taught by Dr. Jo Handelsman, who is shown here in the picture. And she used to actually teach this course called Microbes to Molecules in the year 2012. That was the first year she taught it when she was at Yale University. And this class had about, I think, 6 or 7 students, or just basically a handful of students. And she taught this course while she was a professor there. But then she had to leave Yale to join President Obama at the White House Office of Science and Technology. And when she left, the course kind of continued, but she wasn't there. And when she came back from her tenure at the White House, she came back to the University of Wisconsin, and she set up this thing called Tiny Earth, which was no longer just one course that was being taught at one university. It became what we now refer to as Tiny Earth. And what is that really? It actually now, and there goes my dog, I'm sorry about that. So Tiny Earth actually now is a network. It's a vast network of more than 800 instructors. And we tend to call these instructors Tiny Earth Partner Instructors, or TEPIs. And these instructors are spread out, as you can see in this map, across 33 countries all over the world. And we teach about, all put together, over 16,000 students a year who engage in this Tiny Earth project. So one of the reasons that Tiny Earth kind of came into being is the awareness that there unfortunately exists, excuse me, very pressing global challenges And the three of these are mentioned here because Tiny Earth attempts to address these main ones. Excuse me. The global challenges predominantly are increasing opportunity gaps in STEM higher education. That's one big gap that we see, or one big problem that we seem to have. The other big problem is the increase in antibiotic resistance. And finally, the issue of soil erosion as well. So these seem to be global challenges that are existing currently. So how does Tiny Earth play a role here? So Tiny Earth tries to address these global challenges through a three-pronged approach, basically. So one, Tiny Earth hopes to inspire diverse students to persist in science. The hope is by engaging in the project Tiny Earth, we'll do our part in inspiring a diverse population of students to persist in the sciences. The second aspect is, through this project, we will hopefully address the issue of the dwindling supply of antibiotics and combating antibiotic drug resistance. And finally, third, we will hopefully learn more about our soils and learn how to protect these soils, which are a precious resource for us. So how do we go about doing this? So the first goal being, how are we going to inspire diverse students to persist in STEM? So as I pointed out earlier, Tiny Earth is a cure. So it is a course-based undergraduate research experience. And for those of you who are familiar with this, I apologize, but I'm just kind of quickly trying to go through what the primary hallmarks of a cure are. So a research experience is considered a cure if it can provide a student with an authentic research experience, if it can provide a student an opportunity to see the project go through multiple iterations. It's a cure if the students get to own the project. It's a cure if the project, excuse me, if the project is trying to answer something that is relevant in everyday life. It's a cure if there is some degree of discovery that the student might come up with. And finally, a cure is one where the student gets to disseminate what they're finding with the community that they're in. And what Tiny Earth does is it is a cure, and participating in this cure, the students get to do a little bit of each of these. They get to do authentic research. They get to own their project because it is something they're doing. They're answering a very relevant question. They get to hopefully discover something, and we provide them many opportunities to disseminate the data. It is known, actually, there's been a lot of research that has pointed out that cures are, if a student participates in a cure, it actually promotes student learning. There is research that has shown that cures help increase research skills of a student. They help increase project ownership. They increase the confidence and self-efficacy of a student. And this is actually a study that was done on students who were engaging in Tiny Earth projects. And they noticed that students who were engaged in a Tiny Earth project demonstrated greater scientific self-efficacy and scientific identity. And at the end of it all, it actually promotes retention in STEM. It increases the persistence of students in STEM and keeping them interested in the sciences. The second aspect that Tiny Earth tries to address is the problem of the dwindling supply of antibiotics. So antibiotic discovery was prominent in the early 1950s. And unfortunately, as time went by, there was a drop in new discovery of antibiotics. Often what is quoted is one of the reasons that a lot of pharmaceutical companies shied away from antibiotic drug discovery is because of what's highlighted here in terms of this. I'm just going to try and see if I can get a pointer for myself here. Okay, let's. Okay, never mind. So what's showing up here in the middle is the time it takes to develop an antibiotic by a pharmaceutical company because of the amount of money involved in investment needed in research and drug development, and just the process of going through the clinical trial phases. What is often quoted is it takes at least 9 years to 10 years to get a drug from a discovery, early discovery, all the way out to the market. And what is often quoted by pharmaceutical companies is that it costs around $1 billion to get this drug out to the market. So this is a heavy burden that the pharmaceutical companies feel, and so they probably shied away from antibiotic drug discovery. However, while drug discovery or development of drugs has dropped over time, the use of antibiotics globally has been on the rise. So it's kind of a double whammy where we're using more and more antibiotics, but we're also not developing enough new antibiotics. And what has this led to? This has actually led to one of the major health crises that we seem to be facing worldwide, which is an increase in the antibiotic drug resistance. Basically, this idea that there are more and more antibiotics out there which are becoming increasingly resistant to, our bacteria are becoming increasingly resistant to most of the antibiotics that are out there. That's what's showing on the left here. And on the right, it's just an indication of the many antibiotics that have been discovered over time. And at the bottom of that horizontal graph, it indicates with every discovery of novel antibiotic, we've unfortunately also started seeing a grave increase in antibiotic drug resistance. And as a result, a lot of the antibiotics that are being used right now are not as effective as we'd like them to be. And this goes hand in hand with the fact that there have been no registered new classes of antibiotics that have been discovered since almost the 1980s. So there's this vast void of novel antibiotics and a rapid rise in the number of antibiotic drug-resistant bacteria that are being discovered, and also the number of diseases that they seem to be causing. And in fact, there's a lot of research, and including very recent research and reports that have come out that point out the grave situation that we're in right now. It is predicted, or it was predicted in a recent Lancet study, that there were about 1.27 million deaths in 2019, and about 4 or 5 million deaths where antibiotic drug resistance actually actively played a role in that death scenario. And there are some grave projections which say that we will most likely see a skyrocketing case in the number of deaths that are likely to happen from infections that are going to be caused by drug-resistant pathogens. So what is being done currently? So in fact, in September of this year, the UN had a high-level meeting on antimicrobial resistance, and they've come up with this plan worldwide to commit to actually a decisive action. And they want to approach this from a multi-pronged approach and not just from one angle. They want to look at it from the human health perspective, from agricultural perspective and from animal health perspective. Again, all areas where antibiotics likely play a role. And what their goal is to try and reduce the quantity of antimicrobials being used in general in at least our agricultural or food systems, decrease the number of antibiotics we're using in our veterinary population, animal population, and on the human health side, at least ensuring that the antibiotics that we're using will have minimal side effects, and the hope that we can use antibiotics that will have a lower potential to end up becoming an antimicrobial resistance or end up having microorganisms develop resistance to these antibiotics. And of course, a big aspect of putting this on a global scale is to ensure that we help countries meet these goals by at least providing some kind of financial support from the national level in the United States or globally as well. How do we help countries achieve these goals? So this on a global scale, on a national scale in the US, we're beginning to think about these things. So locally, what is it that we can do? And this is where Tiny Earth comes in and says, here is a part we can all play. And here, what we're trying to help with this, address this antimicrobial drug resistance issue is we're going to utilize our students. With the help of our students, we'll try to address the dwindling supply of antibiotics. How are we going to do that? We're going to get our college students to worldwide to actually start looking for these new antibiotics. And where are we going to look for them? We're going to hopefully look for them, in soil bacteria. And this is roughly how Tiny Earth works. I will go into it a little bit more detail in a few slides, but the idea being we will have our students isolate bacteria from the soil, screen those isolates for bacteria that are actually producing antibiotics. From there, pick up those antibiotic-producing bacterial isolates, characterize them, understand who they are, hopefully extract the antibiotic or compound that the microorganism is producing, and try to identify it and elucidate what this compound might actually be. In addition to this, which is the experimental aspect, Tiny Earth has some wonderful opportunities for their students to participate on this global platform. And in fact, how timely of your organization to host this webinar during what we refer to as World Antimicrobial Resistance Awareness Week, which is basically November 18th through the 24th in the United States. So it's wonderful that we're doing this together this week. And Tiny Earth allows students to be a part of this. And the way we do this is there is a call out actually from Tiny Earth to all students who are engaged in this project to really work together, see if they can, to make it a little fun on the side, see if they can participate in what we call this Every Step Counts, to showcase to the students that the work that they do in their classrooms is actually worthwhile. They are part of this bigger global search out there. And we're asking for these students to participate in this photo contest and raise awareness about antibiotic drug resistance, about the need for antibiotics, and also showcase that our students are participating in these research projects. So it's a wonderful opportunity for all our students to submit a photograph of them doing actual active research in their laboratories, in their classrooms, and it gets sent to Tiny Earth, and they feature it on their webpage. And then there's a contest, and usually one or two of these photographs are picked and shared with the CDC and the World Health Organization, and they get showcased on a global platform. So I think it's a wonderful opportunity that we also provide for students on the side. And the third goal that we have with Tiny Earth is to protect our soils. So this was a book that was recently published by Dr. Jo Handelsman, the founder of Tiny Earth, and she published this book called A World Without Soil. It's a wonderful read if you ever get a chance to read it. It talks about our soil and the need to protect it. And the unfortunate thing with our soil is we are losing our soil at quite an alarming rate world around. And this is not just a problem that is for our country, the United States, but it's a global issue. In the United States, we are noticing that we're losing our soil at about 5 ton per acre per year. And in the state of Iowa, which is actually the second largest agricultural producing state, the acreage is eroding at around 25 to 50 tons per acre per year. We're just like losing that soil. And in fact, it's even more drastic in some years when the weather takes a really bad turn. The loss of soil from some areas in Iowa is in the order of about 100 tons per acre per year. So it's quite stark that we're not just that we're seeing these problems with antimicrobial drug-resistant pathogens coming up and our inability to treat them, but we're also losing our soil. And there's a reason why I'm talking about soil being a big issue, because again, why do we worry about soil? Why is this such a big issue? Is because soil is actually a wonderful resource for antimicrobial compounds. Of course, there's one aspect of soil. We need the soil for agricultural produce to be grown, but the other aspect is also soil does serve a wonderful source for antimicrobial compounds. And in fact, from this picture, what we can kind of see is over 50% of natural products come from soil-dwelling bacteria. And so, in fact, many of the antibiotics that are out there have originated likely from microorganisms, bacteria, or other microorganisms that survive in the soil, and they were actually producing these compounds. And in fact, 1 gram of soil contains over 10 billion bacterial cells. And the reason we point this out to our students often is because when they engage in Tiny Earth as the project, they begin at this stage. They begin with that 1 gram of soil. And it's kind of, it's quite stark for a student to realize that, oh, I have this 1 gram of soil and there could be over 10 billion bacterial cells here. And maybe I'll find that one bacterium here that might be producing some novel compound. And that's the hope and the search that we're on. And that's why we go to soil as the source for our Tiny Earth project. So how do we find new antibiotics from the soil? We literally start by collecting soil and begin to explore. Does the soil have microorganisms firstly? And secondly, if there are microorganisms, do these microorganisms actually actively produce antibiotics? So Tiny Earth is in itself referred to as an antibiotic discovery pipeline. And the hope, yes, at the end of it all is, could we discover a novel antibiotic? But there are many layers to it. Of course, right at the top are the instructors, all the faculty members, the teachers who sit around worldwide, and the teachers get trained on a yearly basis to teach this course, to teach this as a class to their students. The students are on the next level where these are the students who actually engage in that active research. They're dealing with the local soil, wherever they may be, and they're engaging in that area, trying to analyze the soil and the microorganisms from that soil. The students then have to disseminate that data. Part of that dissemination sometimes involves them inputting this data into this large database that is shared among all of us Tiny Earth partner instructors. And this just kind of points out that we have about 26,000 isolates in there. These are isolates, which means these are bacteria that students have found in the soil that they've been studying in whichever part of the world they have been in. From there, once we have the pathogen or the pathogen inhibiting isolate, the microorganism that's producing the antibiotic, we then move to understand what that antibiotic might look like. Now, some of us are able to do this in our labs. If one is in an institution that has the facility to do the chemical analysis, do compound analysis, that's great. But if someone is in an institution that doesn't support that, they don't have the facility to do that, Tiny Earth has what it refers to as a chemistry hub, which is basically a lab and a series of researchers where we can send our samples to them and they will further do the analysis of the microorganism, of the compound that the microorganism is producing, and hopefully from there find that antibiotic or antibiotic structure. So Tiny Earth offers this ability to students to work through this process. There is a student research guide that's provided, and the students learn to critically think through these experiments, walk through these experiments. How would they design some of these experiments to get to the end goal of perhaps discovering something novel? For the instructor, Tiny Earth is a course that is actually based on a great legacy of evidence-based teaching practices that was put in place by Dr. Handelsman and Sarah Miller and Christine Fund. These are people who wrote the book on scientific teaching, on how to teach science to students. And actually that pedagogy is built into the cure. So any faculty member who is engaging in Tiny Earth is trained on this pedagogy, is trained on teaching science to the students. And if you had to walk through and look at how the entire workflow looks, the research workflow, it's broken up into 4 modules. And what's nice about Tiny Earth as a project is it's modular. So each faculty member can pick and choose what they can do. And if there's something they cannot do, they could partner with another faculty member to fulfill that aspect. And we refer to it as Module 1 being the isolation of bacteria from the soil. Module 2 is actually looking for bacteria that are active antibiotic producers. Once you identify those, and again, when we say we identify these antibiotic producers, we want them to be able to produce antibiotics against what we refer to as the escape pathogens or the escape-safe relatives. So the escape pathogens, it's an acronym for the organisms that are notorious out there in the clinical setting, organisms that are highly drug resistant and are also, unfortunately, causing a lot of hospital-associated infections. But we cannot work with those pathogens in our teaching environment, so we work with what's referred to as escape-safe relatives. And when we identify these bacteria from the soil, we want to see, do these bacteria produce compounds that would kill off these escape-safe relatives? And the idea is if they work against the escape-safe relatives, the hope is they will eventually also work against or have antibiotic properties against the escape pathogen. Once you identify that or find one such bacterium, Module 3 involves actually getting to know that bacterial isolate. A lot of faculty members do either very strict microbiological analyses, excuse me, or studies, or one can do genetic analysis on these microorganisms to understand who you're dealing with, which microorganism are you dealing with. And then following that, depending on the expertise of the faculty member and the resources available to the faculty member, they then can go into actually extracting that compound that perhaps this microorganism is making. So this is the entire research flow. most of us who work with our students in our classroom confidently get through module 1, 2, and 3. Usually it's a timing issue in a semester-based system. Sometimes we don't have time to get through to module 4, but oftentimes we do that as extended research with our students. A big key aspect of a CURE or a course-based undergraduate research experience is dissemination of that information that students are finding. And this gives students a wonderful opportunity, and there are multiple opportunities for them. It begins with the discovery database, where students are taught how to enter data into a database. That faculty then we all have access to it, and we can look across. So for example, I could compare, I could say, here I'm in South Florida, my students are looking at soil in South Florida, and we could then say, I wonder how soil in Puerto Rico looks. I wonder what students in Puerto Rico are finding. And we could actually go to the database and start looking. And it makes for nice small projects, comparisons that our students could work with each other to do a quick analysis of what are you seeing versus what am I seeing. There's an opportunity to also send microorganisms to the chemistry hub at Madison, Wisconsin, where the chemistry hub exists. Tiny Earth has an international symposium that they host every year, where students come and they can present their work, what they have found, what they have done at their universities. And yes, there is also opportunity for publications. Oftentimes, faculty members and students write articles, write journal articles about the work that they've done or things that they have found on their campuses. There are some examples here of just Tiny Earth publications that either people at headquarters have put together, beginning very much with Dr. Handelsman's work in science about STEM diversity and how we can fix the classroom through these kind of innovative hands-on experimentation with our students, to a variety of articles that faculty members and students have written about the experiences they've had in their classrooms. While there's a lot of advantages for students, being part of Tiny Earth offers a great resource, is a great resource for faculty members, because getting trained to conduct Tiny Earth allows instructors really to engage in novel pedagogy. Faculty members are trained on adopting inclusive classroom practices. We're trained on scientific teaching principles that have shown to work better and have been shown to improve STEM retention. So faculty actually get trained in all these aspects, and that's a great way to bring about a change in our classrooms. Tiny Earth, of course, isn't just restricted to one area or one location. We often try to expand our reach. A big way we do that is through what we refer to as Tiny Earth Partner Instructor Trainings. And as I mentioned, it's based on scientific teaching, mentoring, and of course, a little bit about the actual experimentation. And this is just a sample of what the Tiny Earth Partner Training looks like. Oftentimes, when faculty attend this training, we work in facilitated groups that are, we kind of get grouped together as faculty. We learn from each other. There's a lot of mentoring that goes on from other faculty members who've done Tiny Earth in their classrooms already. We dive deep into the content of scientific teaching, how we could adapt Tiny Earth to our particular classroom, understanding very well that every scenario is different, every college is different, every classroom is different. But Tiny Earth does a wonderful job of helping fit it into any type of classroom. As evidenced before, we've done this in multiple classrooms around the world where colleges and students and faculty look different, and the environment is very different. Of course, we learn scientific teaching principles. How do you teach science effectively? That's a big process of it as well. and using evidence-based approaches to relay that scientific content, learning to make it more learner-centered. And when we attend these trainings, it's really very hands-on. We get to walk away with a lot of our courses built and neatly designed around this principle. Of course, we also go through the lab research and protocols. For microbiologists, the microbiology aspects may be very simple and come easy, but then there are other modules. There are chemistry modules in Tiny Earth that one could learn about. There is genomics that one can pick up. So there's all these avenues that the research can go down. The biggest help that I find, or the biggest thing of importance for me, is this network of trainers and facilitators and mentors that we gain through this group of about 800 instructors worldwide. We all become sounding boards for each other. And being part of Tiny Earth also offers you many opportunities to build that leadership capacity to then go on and become a mentor for someone else, or go on and conduct a training in a different location, go on, present your information in a different location. There are advisory board members. I want to kind of just point out who are all the people who make up this Tiny Earth group. It's stood and risen on the shoulders of Dr. Joe Handelsman, but there have been a lot of other people who've taken it forward. Dr. Nicole Broderick, Dr. Deborah Davis, Dr. Enid Gonzalez Orta, Dr. Joanna Klein, Dr. Christian Labby, all these people who have contributed in different ways towards Tiny Earth over time get to be involved and become part of committees and work together. And of course, the core staff, which is made up of course, our founder, Dr. Joe Handelsman, and the executive director, Sarah Miller, and all the other folks who participate in University of Wisconsin at Madison sit at headquarters, which is in Madison, Wisconsin. And they also have a huge support team that helps faculty members digest the content, helps our students disseminate the content out onto the database, or understand the lab protocols, or make changes to these lab protocols. This is the entire team that's part of it. There's a lot of opportunity for faculty members to do professional development, from workshops to webinars to writing content to publishing content, and of course also engaging with the community in different forms or different ways. Tiny Earth is constantly evolving. There's new content that's constantly coming out. A lot of faculty members can also get involved in writing some of this new content. And in fact, there's new curriculum that's constantly coming out, written by fellow Tiny Earth partner instructors. There are fellow partner instructors who designed the chemistry module, who designed the metagenomics module. And there's also a lot of opportunity to get involved in other research that faculty members are involved in, whether it's failure studies or soil research. There's opportunity that we as partner instructors can get involved in those research projects as well. I have to thank all the diverse portfolio of partners that help Tiny Earth happen, from the University of Wisconsin, the Wisconsin Institute of Discovery, to the Howard Hughes Medical Institute, to NSF grants that help and support the Tiny Earth partner instructor training events that happen in Wisconsin and at different places. And also, these are the organizations that help host all the symposia that happen on an annual basis. I finally want to thank my local community, Nova Southeastern University and the Department of Biological Sciences, that has always supported me in helping bring Tiny Earth to our institution and help support me in running Tiny Earth and involving my students at our local university. A big shout out to all the Tiny Earth partner instructors whom I've worked with over time. The Tiny Earth leadership team in Madison, Wisconsin, all the committees I've been part of. And of course, a big thanks to Dr. Joe Handelsman, whose philosophy of scientific teaching is what drew me to Tiny Earth, and I am really proud to be a part of this organization. That is all I have. I can stop here and take any questions.
Thank you, Dr. Raja, for a great presentation on Tiny Earth. In fact, I'm not sure if there are other scientists in Puerto Rico working with Tiny Earth, but one of my colleagues, Lilian Casillas,
Yes,
she was really into Tiny Earth, and I remember when we saw her proposal, and it was really, really fascinating to see. And I know students that got involved in her work directly at the general microbiology lab. So it
right.
was fascinating.
Right,
But
absolutely. There are actually several faculty members out of Puerto Rico at different institutions where they have worked with Tiny Earth over the years, but there's now again a renewed interest, I want to say. And I think, if I'm not mistaken, there are a few faculty members who are getting trained in January at one of the training sessions to take back Tiny Earth to some locations in Puerto Rico. So we're really excited about getting more faculty from Puerto Rico involved. And I think it'll be wonderful to see more people engaged in this.
yes, I'm checking if there are any questions in the chat, but before someone writes a question or raises their hand, I also have a comment because precisely on during this week I have been reading about the model of educate, advocate, act now. That's really so powerful. But at the same time, news from war-torn places and places with social unrest, the news are very alarming regarding staff, regarding pseudomonas, regarding other infectious diseases which are rampant.
Yeah.
And doctors are using like vinegar or something to try to cure the wounds. So really, how can microbiology and microbiological research sort of like push, make public, increase public health issues concerns? Because I think that many people are living like in the limbo. They don't know what's happening.
No, absolutely. You bring up a wonderful point. And in fact, I come from the country India, where antibiotics are very easy to procure. And oftentimes people just get antibiotics without prescriptions, and they're using it for a disease that it's not meant to be used for. So this becomes a big issue. One of the things that Tiny Earth does is they do a big push in the public health space, utilizing students to kind of get the word out in new ways. I'm more, I guess, based on my age, perhaps, I'm thinking, okay, we should write a paper, do the research, publish a paper, get this information out there. But maybe we need more novel methodologies to get the information out there to people who need it. And there are new campaigns out there that students and people who are involved in Tiny Earth are trying to do, utilizing things like social media to get information out there, educate the public about these problems. In fact, there are some Tiny Earth partner instructors who hold their class dissemination poster presentations. For example, they do it at the local library, or they do it at the local art museum. They kind of hold these events at different locations just as a way to, as you pointed out, get the public more involved. In fact, one of the biggest conferences that Tiny Earth does is at Green Bay in Wisconsin. They actually hold it in a football stadium, which is like this big revered football stadium for the American football, and they invite the entire community. So about 500 people show up to this event, and it's nothing but students presenting what they did in their classroom and why this is so important, why we need to worry about antibiotics and drug-resistant bacteria. So there are all these new ideas that partner instructors, there's just faculty are coming up with, and we're all trying to learn from each other and see if we can do these new ways of disseminating information.
Okay. Any other questions for Dr. Raja, please? and Natalie, Valerie.
Thank
We
you.
have one comment here. Great project, Dr. Raja.
Thank you. We'd love to see, we'd love to have more faculty involved. Like I said, I think there's a few people who are getting trained, a few faculty. There are some faculty already involved. I think someone has their hand.
Yeah. Angelica.
Yeah, good evening. Thank you so much, Dr. Raja. It was very informative. I don't know if you mentioned it, but does Tiny Earth also help acknowledge and inform high school and middle school teachers? Because I know that in Puerto Rico, more academic university professors want to power them so that they can help bring up the next generation of undergraduate students and future scientists. So I guess I just wanted to know more about that.
Sure, absolutely. So there are some faculty members who, or rather teachers from high schools who have been trained and have taken Tiny Earth to their high schools. Some of us faculty members actually coordinate with a local high school that's near me. For example, I'm actually on a campus, a college campus that also has a high school on my campus. So I actually reached out to the high school faculty member, and they were really interested, and we kind of paired up with each other. And I have my undergraduate students go talk to the high school students, and it works as a wonderful mentoring project. So there are those opportunities, absolutely. We do try to involve high school students, high school faculty members, high school teachers as well. And there are multiple ways to do it, and Tiny Earth encourages that. Some schools are a little more difficult. I know, for example, the public school system in the county where I am, they don't have microbiology being taught on their campuses. They don't like us bringing in bacteria on plates to the campus. So it becomes a little harder then to actually do experiments, but we definitely have gone and given talks at their schools. So absolutely, we encourage that. And if there is an opportunity, there are ways it can be done. And we have ideas around how some of us have done those things.
Thank you so much. And if we would want to contact you, I know that Dr. Blues put in the chat that if we can get your email
Absolutely.
Okay.
Yeah, it's on my slides, and I think they'll post my slides at some point somewhere. Yeah, feel free. I'll put my contact here in the chat as well. You can email me. Absolutely happy to share any information, and also happy to put people in touch with the specific faculty perhaps who are coming in from Puerto Rico who may have already started the project. And again, it's always nice to get people from different parts of the country as well. I know Puerto Rico may have universities and colleges in different areas, and who knows, soil's probably different in each of these areas with your very diverse like nature and landscape that Puerto Rico has. I'm sure of that. So that would be a nice project in itself to kind of compare what's happening across the island.
Thank you so much for your comments and answers to the questions. So anything else? Natalie? She was our host originally. She was recording, so I'm waiting for it.
Yeah,
Thank you so much for
thank
the
you so
opportunity.
much for your time, Dr. Raja.
My pleasure.
We thank everyone who connected today, and for Dr. Anna for being our moderator as well. So have a good night, everyone.
Thank you. Thank you all.
Thank you.